Hi, my name is Alexandra Dickey. I'm a student in the RAP program here at WVU, and I worked with my mentor, Dr. Julie Brzezinski-Lewis, on her study short-term behavioral, physiological, and brain differences in real-life and virtual reality social interactions. Human social interaction can tell us a lot about the brain based off the response, especially when the conversation has a different tone to it. So for example, if you are in a stressful situation, your brain and body are going to give off different responses than if you were talking to someone calmly. Virtual reality is a new technology which has the unknown efficacy in human research regarding social interactions. VR has the potential to be very beneficial to both medical and physiological settings with the ability to put patients in controlled settings and face-to-face environments. The aim of our study was to test physiological responses in face-to-face versus virtual reality stressful interactions, and our hypothesis is that we predict no statistically significant difference in physiology for a stressful face-to-face interaction versus the same interaction taking place in virtual reality. For methods and materials, we chose two different scenarios that we were going to put our subjects under. The one is confronting a messy roommate, and the other is confronting an uncooperative group member. These interactions were going to be delivered via a live setting or through virtual reality using an actor. The actor featured is the one on the left, that's Helen Melnick, and as you can see, she has the avatar that was being used in the virtual reality setting. We conducted the virtual reality interactions via the Samsung virtual reality headset and through the app VTime, which allowed the face-to-face interactions. The blood pressure and heart rate were measured for 52 subjects before and after during each interaction. The order of stressful scenarios and VR versus live was randomized, so each subject received a different one at each time. Data was analyzed by grouping each participant into one of four groups: virtual reality mess, virtual reality group, the live mess, or the live group. A paired t-test was conducted for each physiological measure using JASP, which is a software program that we use to find our t-test values and our p-values. If you have time to pause, please read the script that's based off of our group project scenario. The confederate would read the script out loud, and our subjects, either via a live interaction or virtual reality one, would have to respond to them, and this is what induced the reactions in their brain. Here are our results based off of all 52 subjects and based off their systolic and diastolic blood pressures. There's no significant difference for all data for mess scenario, but there is, however, a significant difference seen with systolic blood pressure for the group's project scenario. Here are some graphs that we created based off of the measurements and the results that we got from all 52 subjects after completing and analyzing all of the data. As you can see in graph A, it's the mean systolic blood pressure of all the data combined. That means that it's the VR virtuality group and mess and the live group and mess systolic blood pressure. As you can see, there's not a very large significant difference between rest 1, rest 2, and event 2. However, there is a little bit more of a significant difference between virtual reality and live for event 1. This is based off of the graph seen to the right, the mean systolic blood pressure of the group project. This graph B, the mean systolic blood pressure of the group project, is where there is the significant difference seen in the p-values in the chart. As you can see, there's a very large difference between the live and virtual reality results of event 1. Graph C is the mean systolic blood pressure of the mess scenario. There's not really a big significant difference found in this, which is further supporting our hypothesis that there should not be a significant difference between the results of the virtual reality and live interactions. If you have time to pause, please do. So our conclusion is that although our hypothesis was supported with our data, the significant difference observed for systolic blood pressure, which was greater in the live and virtual reality conditions, suggests caution in needing to assume a virtual reality experience is just as real as a live interaction. Although it can be very useful and you can control the variables much more tightly, there are, however, some differences, obviously, because it's not a face-to-face interaction as a live one would be. However, the potential to increase in response for some live interactions may open up another avenue of study as to what aspects of live interaction make it more enriching, which is a really relevant question for today's video game and currently Zoom-dominated world due to COVID. Given these circumstances, virtual reality could prove to be a very useful tool that can be used in many different circumstances. For future directions, our lab is currently working on a mobile PET scanner, which would provide and could enable live imaging during more natural social interactions. Virtual reality could also be potentially used in collaboration with the PET for future research in social interactions. This is also an example of the prototype of the mobile PET scanner that we would be using in all future subjects. This goes on your head like this, and then this would be scanning everything. And we actually have even built a mobile PET scanner that can be used for patients in hospital beds. So the mechanism is basically that this can move up and down, and then this is what reads everything.